Diabetes is usually understood as a disease that develops over time, shaped by lifestyle, immunity, or aging. But in rare cases, it appears almost as soon as life begins. Some babies develop dangerously high blood sugar levels within weeks of birth, long before diet, environment, or immune attack could play any role. Hi, my name is Soumya Pillai. Welcome to another episode of Pure Science. For years, scientists have known that genetics must be responsible in many of these cases, yet the exact biological failures behind them have remained unclear. New research from the University of Exeter has now identified a previously unknown genetic form of diabetes in babies, revealing a fundamental breakdown in how insulin-producing cells function. The discovery comes from the study of 6 infants from unrelated families who all developed diabetes before the age of 6 months. Their condition did not fit any known genetic category of neonatal diabetes, prompting researchers to examine their DNA in greater detail. Using advanced genome sequencing, the team identified damaging mutations in both the copies of a gene called TMEM167A. Because both copies were affected, the gene could no longer perform its normal function. This established TMEM167A as the cause of a newly recognized recessively inherited form of neonatal diabetes. To understand how this genetic fault leads to disease, it helps to look inside the pancreatic beta cells that make insulin. These cells are among the hardest working cells in the body. They continuously produce insulin, fold it into the correct shape, package it, and release it precisely when blood sugar levels rise. This entire process depends on an efficient internal system that manages protein production and transport. TMEM167A plays a critical role in keeping that system running smoothly. When the gene is defective, the machinery inside the cells begin to break down. Researchers recreated this process in the laboratory using human stem cells that were turned into pancreatic beta cells. When TMEM167A was disrupted in these cells, insulin started to build up inside them instead of being properly processed and released. This buildup triggered severe stress in the endoplasmic reticulum, which is the part of the cell responsible for folding and quality controlling proteins. Cells can cope with limited stress, but when it becomes overwhelming, they activate self-destruct pathways. As beta cells begin to die, insulin production dropped sharply, leading directly to diabetes. What makes this newly identified form of diabetes especially striking is that its effects are not limited to the pancreas. All of the children studied also showed neurological symptoms, including developmental delay, epilepsy, and unusually small brain size. This connection is explained by the fact that TMEM167A is active not only in beta cells but also in neurons. Like beta cells, neurons rely heavily on precise protein handling to function normally. When that process fails, both brain development and insulin production are affected, linking metabolic disease and neurological symptoms through a shared cellular mechanism. Clinically, the discovery has important implications. Diabetes diagnosed within the first 6 months of life is already a strong signal that the condition is genetic rather than autoimmune. But without a clear genetic explanation, families often face long periods of uncertainty. Identifying TMEM167A as a causal gene expands the list of targets doctors can screen for when evaluating newborns with unexplained diabetes. A precise diagnosis allows clinicians to recognize early that neurobiological complications may be part of the same condition rather than separate problems. This awareness can shape long-term care. While insulin treatment remains essential to control blood sugar, clinicians can also monitor affected children more closely for seizures, developmental delays, and brain growth abnormalities. Genetic confirmation of the condition also enables more accurate counseling for families. Because the disorder is inherited in a recessive pattern, parents may be unaware they carry the gene. Understanding this helps families assess the risk in future pregnancies and provide clarity that would otherwise be missing. Beyond this immediate clinical relevance, the study has wider implications for diabetes research. Stress and failure of insulin producing cells lie in the heart of many forms of diabetes, including those that develop later in life. By identifying a gene that causes beta cells to collapse under the pressure of insulin production, the research highlights a biological pathway that may be relevant so far beyond this rare condition. Rare genetic disorders often act as windows into fundamental processes that operate in more common diseases. Published in the Journal of Clinical Investigation, the findings confirm that there are a lot more mysteries around diabetes that we still do not understand. While only a small number of children are affected by this newly identified form, the study deepens scientific understanding of how insulin production can fail at the cellular level, even in the earliest days of life. That was all from me. I am Soumya Pillai, and you were watching Pure Science on the Print.